The first objective of the lecture, we are going to look at xylem and transpiration. Now, plant, they will imbibe, they will absorb, they will take and transpire water, meaning to release water more than what animals and humans do, as they have no recirculation system. And it is about 99% of all water which entering the roots leave the leaves via the stomachs through the process of transpiration without ever taking part in any metabolism. In this case, metabolisms in plant, we are referring to photosynthesis or any other metabolism which may take into consideration of a process called catabolism or anabolism. But the facts that we have to know, plant which imbibe and transpire water more than what humans and animals do. And almost all of the water that they imbibe and transpire leave the plant through stomata of what the process they call transpiration. In a single plant, they can transpire almost or up to 60 liters of water in one growing season, regardless of any types of plant. Water movement in plant is actually due to the differences in potential between soil, root, stem, leaf, and atmosphere. It means that the water capability or the capability of water to move, to transport, to crawl from soil to the root, to the stem, to the leaf, and out to the atmosphere, it is because of the differences in potential in the respective place. Under normal condition, the water potential in soil is higher than in root cell, resulting in water will flow to follow the potential gradient. This is known as bulk flow, and this is the primary force that drives the water through xylem. I repeat again, under normal condition, the water potential in soil is always higher than in root cell. It will result with the water from soil that will flow, following the potential gradient into the root cell. This is known as bulk flow. And this is the primary force that drives the water through xylem from the beginning. The aqueous solution of dissolved mineral in the xylem is known as sap. Remember, any aqueous solution or dissolved mineral in the xylem known as sap. Now, the bigger question that we have to ask here, is it the water being pushed or pulled throughout the whole system in order for the plant to do transpiration as well as translocation? And this question will be answered in the next following slide, whether the water is being pushed or pulled, or the water are conducted in both mechanism, which is push and pull in order to fulfill the requirement of transpiration and translocation. As what we can see from this diagrammatic feature, it is showing there are two highlighted circles which on top showing the percentage of water transpired and the lower circle showing the percentage of water absorbed. Now, let's look at it one by one. On the lower circle, we can see the water flow through root hair. Yes, root hair is the most effective, efficient, and significant root structure which help to absorb, to imbibe water from the soil into the plant. And we know that almost more than 95% of water absorbed by root hair, and the remaining 5% and below absorbed by the other parts of the plant, including endodermis. Meanwhile, on the top circle, it's showing the percentage of transpiration through stomata. We know that 99% of the water absorbed by the plant, they will release in a form of water vapor through stomata in a process called transpiration to atmosphere. Only less than 1% of this water transpire out to the atmosphere through the other parts of the plant, including lenticel in woody plant. So we know that plant, they don't have a recirculation system as what the animal and humans do. Therefore, almost all of the water that they absorb will return to the atmosphere. In this slide, I'm sharing with you the function of some of the important structure in the plant in order to conduct the process of transpiration. In roots, the mineral ion which dissolve in water will flow into the root by apoplast or vacuolar pathway. I will explain in detail what is apoplast pathway and vacuolar pathway and how the apoplast pathway and vacuolar pathway are being conducted to transport the mineral ion dissolved in water through roots. Meanwhile, in endodermis, this mineral ion secreted actively into xylem vessel. Yeah, we know the facts that not only root, especially the root hair, but the endodermis also conduct the flow of water as well as the dissolved mineral. But the mechanism of how the water flow through endoderm compared to root is definitely different. In xylem vessel, the mineral ion which dissolve in water will be carried to site where they are used by mass flow in the transpiration stream. Mineral ions diffuse or actively transported into meristematic cells. It can be young leaf, it can be to flower, fruits, or any storage organ. All of these are actually involved in the process of translocation, which conducted by phloem. The facts that we have to Remember, roots, endoderm, xylem vessel, they transport water, but root transport water from soil into the plant system. Endoderm transport water from the soil into the plant system. Meanwhile, xylem vessel, they are transporting water within the plant system. In order for us to further understand about how xylem conduct the water throughout the whole plant system, as well as the whole transpiration process to take place, it involves the process of absorption of water, absorption of dissolved mineral ion in water from the soil into the root cell. Now, therefore, I suppose we have to look at the mechanism of absorption and transport of water and mineral, especially in the cell membrane, because it involves the transportation of this water and mineral ion not only in the root cells but also to the other cells in the plant system. Mineral ions are needed for the plant's metabolism. As I've mentioned before, the most important metabolism in plant is photosynthesis. Without water, the plant are unable to perform photosynthesis. Solutes tend to diffuse down the concentration gradient. This is a very common and very popular and the most easiest way of transporting the solutes in the plant, where the movement or the flow is diffused down the concentration gradient. They do not need the requirement of energy. This is the simplest way. This is the easiest way. Root hairs increase the surface area for absorption. As I mentioned also before, root hair functions to increase the surface area for absorption. Almost 95% of water as well as dissolved mineral ion will be absorbed through root hair, and the remaining 5% and less absorbed through the other parts of the plant. Surface cell membrane and tonoplast of root cell have transmembrane protein. Now, by looking at the words transmembrane protein, we can guess the words trans mean to transport, or as a transportation through a membrane, and they are a molecule of protein. So, these transport protein, they are actually embedded in the surface cell membrane, especially of the root cell. Transmembrane protein, they may function as a channel, as a carrier, or even a pump to move solutes into the cell, out of the cell, or even from cell to cell. So I repeat again, these transport protein are known as transmembrane protein, which embedded on the surface cell membrane. They may function as channel, a carrier, or a pump to move the solutes into the cell or out of the cell or even from cell to cell. These transmembrane proteins, they may function to accelerate, to speed up, to help the movement of solute faster. When we talk about transportation, there are two types of transportation, which is passive transport and also active transport. In passive transport, they are osmosis. Osmosis is a process of transporting through diffusion across a membrane. Generally, they are a slow movement unless solutes travel through transport protein. As I mentioned before, unless the solute travels through this transport protein, which can be either as a channel, as a pump, or as a carrier to speed up the transportation of solute through the membrane of the cell. Active transport, they require energy to move solutes because this type of movement or flow is up to concentration or charge gradient. That's why they need energy, because the movement is against the concentration and charge gradient. Giving you example in the diagrammatic below. Proton pump will create membrane potential, and the potential energy can be used to perform this cellular work by transporting hydrogen ion from cytosol into the extracellular of cell. But they require energy because the movement is up or against the concentration and charge gradient. And the second example, the membrane potential also provides energy to uptake some of the mineral, for example, potassium ion. So, in active transport, either they can create membrane potential or they can also provide energy to uptake the minerals into the intracellular of the cell. In the third example, the membrane potential can also provide the energy for co-transport of ions up their concentration gradient. As in this example, the hydrogen ion move down the concentration gradient. And in the fourth example, the membrane potential also provides the energy for co-transport for some of neutral molecule, for example, sugar, up or against the concentration gradient, but at the same time, the hydrogen ion are moving down the concentration gradient. Therefore, in active transport, there are four types of movement, including providing the energy for co-transport to move up or against the concentration gradient, as well as providing the energy for co-transport for some of neutral molecule, also up or against the concentration gradient. The diagrammatic features that I'm sharing with you in this slide is actually to show you the complete structure of roots, especially where is the root hairs located. As what we can see here, root hairs are developed throughout the taproot. That's why the function is to increase the surface area for water as well as mineral ion absorption into the plant will be conducted effectively. Meanwhile, on top of the slide, I'm sharing with you the basic structure of plasma membrane. And the most important thing, we have to locate the position or the location of the transmembrane protein, because this type of transport protein really works, really helps, and really function in transporting of those ions, mineral ion, or even a neutral solutes such as sucrose across the membrane, either into the cell, out of the cell, or even from cell to cell. We have come to this section in knowing root pressure which resulted with pushing the xylem sap. If you still remember, in the earlier slide, I have posted a question whether the water is being pushed or pulled. So to answer the question, we know that water being pushed. In order for them to follow the transpiration process. And this pushing action is because of root pressure. You have to remember water being pushed in the xylem sap to follow the transpiration process, and the process of pushing is because of root pressure. Water enters the root because the water potential of the root tissue is almost always lower than the soil, with its high dissolved mineral content. This is the reason why the water enters the root tissue. It is because the water potential of the root tissue is always lower than the water potential in the soil. Therefore, the movement of water is following the potential from high to low. Water entering the stele by traveling via one of the three routes, which I will explain to you in the next few slides. Stele here, we are referring to the cortex area, the endoderm, and also the vascular cylinder, which represent the important vascular stream in outbringing the water from the root to the stem and finally to the leaf to be transpired out to the atmosphere. As what we can see from the diagrammatic features showing in this slide, we can see that they are the roots where the water flow from the soil next to the epidermis, next to cortex, next to endoderm, and finally to the vascular cylinder, to be specific, to the xylem. And in this diagram also, we can see the structure of root hair, which I've explained before, the most important structure to absorb water besides epidermis. And they are plasmodesmata, they are Casparian strip, and they are pericycle. Within this stele, I will explain to you what the plasmodesmata do on the cell membrane and what the Casparian strip do on the endoderm membrane, as well as the pericycle in covering the xylem. The three roots that I've mentioned in the previous slide, which flowing or which transporting the water in from the soil area into the xylem, they are apoplastic root, symplastic root, and vacuolar or transmembrane root. This root conducting water movement in different way. In apoplastic root, soil solution is taken up by the hydrophilic wall of the root hair, which provide access to the apoplast, and water and mineral will diffuse into the cortex along the matrix of the cell wall. So remember, in apoplastic root, the soil solution or any dissolved minerals ion is taken up by the hydrophilic wall of the root hair. And this water and mineral will diffuse into the cortex along the matrix of the cell wall. Meaning that the transportation of water and mineral ion is along the matrix of the cell wall. What about symplastic root? The minerals and water will cross the plasma membrane of the root hair cell and enter the symplast, which is enter the cytoplasm, which form via plasmodesmata. That means in symplastic, the transportation of mineral and water, they are crossing the plasma membrane and also crossing the cytoplasm, which form via plasmodesmata. Meanwhile, the vacuolar or transmembrane route, the water and mineral may travel along the apoplast, which are actively transported into the protoplast of epidermal and cortical cell, and then they move inward via the symplast. It looks like the vacuolar or transmembrane route is the most hardest, is the most barrier compared to symplast and apoplastic route. This is because in vacuolar and transmembrane route, water or any dissolved mineral, they have to travel along the apoplast as well as the symplast route. In general, we can conclude that the most hardest way for water and mineral ion to travel is through a vacuolar or transmembrane route, followed by symplastic, and the most easiest is through apoplastic route. As what you can see in the diagrammatic features provided in this slide, there are apoplastic, symplastic, and vacuolar with the distinguished color. So let's recap. The route which transporting water or even dissolved mineral ion in the plant system, especially from the root, especially from the soil into the roots. Next to the xylem. Apoplastic root, symplastic root, or vacuolar and transmembrane root. You may also want to call it apoplast pathway, symplast pathway, or vacuolar and transmembrane pathway. In apoplast pathway, they involve passive diffusion. In symplast pathway, they involve active transport. And we know apoplast pathway is the most important route to transport water and dissolve minerals ion. This is because the easiest, the simplest, and the fastest way. Water moves across spaces between cellulose fiber in the cell wall. We know apoplast pathway involving the water movement across spaces between cellulose in the cell wall. Meanwhile, symplast pathway, the water have to move through cytoplasm of the cell. And the cytoplasmic strand in the plasmodesmata will allow water to move between cytoplasm from one cell to another cell down a water potential gradient. And last but not least, the vacuolar or transmembrane pathway, the water have to move through vacuole, cytoplasm, or even cell wall. It shows that this is the hardest, this is with the most barrier before water reach into the xylem. Besides transmembrane protein, which we know embedded in the phospholipid bilayer or membrane cell, and they are another class of transport protein, which they call it integral membrane protein. However, this type of transport protein is somehow a little bit different in terms of the purpose of them in the context of water and dissolved mineral ion transport. Under the integral membrane protein, there are two types of them, which are aquaporins and suberin. Both are plays a very distinctive roles in transporting the water and dissolved minerals ion. By looking at the aquaporins, we can actually guess the words aqua, which mean water, porin, which mean pore, meaning that there must be something to do with a pore which will allow the water to flow through. Aquaporin is a water transport channel that speeds up movement of water in plant cell. By looking at the diagrammatic features showing in this slide, we can see this aquaporin really helps to speed up, to increase the volume of water to pass through the cell membrane, either from intracellular to extracellular of the cell, or from extracellular into the intracellular of the cell. They are somehow related or similar to the transmembrane protein as a channel, as a carrier, or as a pump. Meanwhile, suberin is a waterproof waxy material which impregnated at the cell wall of endodermis, and the layer of this cell is called Gasparian strip. We have to bear in mind, suberin is only found in the cell wall of endodermis, which is the one cell thick which cover or surrounded the pericycle as well as the phloem and xylem in the middle of the area. So suberin, it is a waterproof waxy material, and this layer which impregnated at the cell wall of endoderm is called Gasparian strip. So what is Gasparian strip, which made up of suberin at the endodermis cell layer? So Gasparian strip, they block, the function is to block interstitial entry of water or mineral passing through along the cell wall at apoplast root into the stele, meaning that when the water through apoplast from outside of the area to passing through the endoderm, they are not allowed to pass through the cell wall. They have to go through the cytoplasm area, meaning through the symplastic root, before they move into the xylem. Thus, water will move across endoderm via the plasma membrane by symplast root and Passac shell before they can get into the xylem. Endoderm and living cell in the stele will discharge water and mineral into their apoplast. The xylem vessel then transport the apoplastic water and mineral upward into the shoot system. To cut a long story short, Gasparian strip function to block any interstitial entry of water and mineral which passing along the cell wall. They are now instructing the water and mineral to go through the symplastic route. The purpose of having Gasparian strip which block the interstitial entry of water and mineral passing along the cell wall, this is because to ensure all of the water, all of the dissolved mineral ion are passing through the cytoplasm of the endoderm. This is to ensure the endoderm will becoming the first layer of screen, the first layer of filter. Only the good material, only the good amount or sufficient amount of water to be passed through into the next level of pericycle cell and finally to the xylem. On top, the purpose of having Casparian strip is to ensure the structure which surrounding the pericycle and also the xylem is nourished and hydrated enough, because we know that xylem is doing their job in transporting water all over the time. Therefore, they need a significant and relevant support from all over the surrounding cell. In a normal day for a plant, it will involve a daytime and nighttime. There must be a distinguished difference between the way the plant reacts during the daytime as well as the nighttime. Let's look at the example of what actually happens to the plant at nighttime. At night, the stomates are usually closed. And this is to prevent leakage of ion out of the stele into the other tissues indirectly. Actually, the stomates closed is to prevent the loss of water through transpiration. And the endoderm as well prevent leakage of ion out of the stele into other tissue. We know that stele composed of all the vascular system. Meanwhile, the other tissues we are referring to the pericycle as well as the endoderm. It will result in the decrease in water potential in the stele due to the accumulations of ion. And this will also subsequently will result in flow of water from the cortex into the stele. And this will cause root pressure, where xylem sap will push up the xylem because of incoming water from the root. And the root pressure, which initially exerted from below, known as positive pressure potential, since the potential increases as one moves up the stem. And this root pressure is sufficient enough to push or to lift water a few feet above the ground. Let's look at it again. At night, when stomata close, no more water in the form of water vapor escape out to the atmosphere. But at the same time, the endoderm will prevent the leakage of ion out of the stele into other tissue, and this will result with the accumulations of ion, and it will cause the decrease in water potential, because in the stele they are high concentrated of ion. Therefore, it will invite water to flow in from cortex into the stele. And this condition is actually producing root pressure, where the xylem sap will be pushed up the xylem because of incoming water from the root. And this root pressure is known as positive pressure potential because they are loaded with water and they have to be exerted from below. And this pressure is actually sufficient enough to lift water a few feet above the ground to the stem and finally to the leaf. As what we can see from the diagrammatic features below, sharing in this slide, we can see the condition or the structure of stomata when they are open and when they are closed. When the stomata open, look at how the physical structure of gut cell. When they are open, the gut cell will swollen and turgid. When the stomata close, the gut cell will shrunk and flaccid. I repeat again, when the stomata open, the gut cell will becoming swollen and turgid because more water influx in. But when the stomata close, the gut cell will shrunk and flaccid because the water are not influx in. I believe you have heard the word guttation, or in Malay we call it embun. We can normally observe or see gutations in the early morning, especially during the high humidity day. Now, actually, guttation is the direct effect of when stomata are closed at night, but at the same time, there are water flow into the xylem structure. Now, let's look at how actually the production of guttation or embones occur because of the flowing of water by the root pressure at night. This guttation or embone, we can actually observe or see them on the small plant. We can't see them on a big plant because they have a special mechanism to extract out or to release water by a structure called lenticelle, where they can find it, where they are located mostly all over the plant stem. Not like a small plant, they don't have lenticelle to make sure the water can escape at night time when the root pressure are pushing up water in bulk at night. Now, in a small plant, root pressure could result in potentially harmful water pressure which has built up at night when the stomata are closed. In many herbaceous plants or in many small plants, they have special opening on the leaf margin which called hydatodes. These hydatodes allow root pressure water to escape, and this root pressure water escape will form a little bit of dew overnight and preventing cell rupture due to too much of water pressure. This process is known as guttation, and its results are generally observable only in the early morning when humidity is relatively high. So now we know what is guttation. So the guttation is a process of producing little bit of water droplet at night. In Malay we call it embun, through a structure called hydrotot. This is because to ensure or to allow the root pressure water to escape. Otherwise, the cell may be ruptured because of the full pressure when the stomata close at night. I'm sharing with you in this slide a few photographic features showing guttation at the hydrotot of the leaf, especially on the small leaf. This phenomenon occurs when the root pressure is strong at night due to high humidity, which causes the exudation of water droplet through hydrotot at the edge of the leaf. So this is the beauty of each of the structure that we may never know before. The plant, they have to survive. They have to mitigate in any unconditional situation. Therefore, they have to adapt with the surrounding. For example, in this case, at night when the stomata close, therefore there are high concentrations of ion in the stele, which inviting water to flow in. It will cause a root pressure which can actually lift up or transporting water into the xylem. But this condition will definitely causing pressure and turgor to the cell. So in order for the plant cell to release the turgor and pressure, therefore the creation of guttation will occur. So once again, before we end this section, let's do a recap of water transport in plant roots. The characteristic of the root itself, which actually relevant and efficiently transporting the water. The root hair with thin cell wall, and they also creating a large surface area over volume ratio for more absorption of water and dissolved mineral. We know that water may transport into root hair, next to cortical cell, and finally to the cell, and from cell to cell. But what makes water can move into root hair? It is because in a normal daily situation, the water potential in soil is always high than the root hair. Therefore, it permits the movement or the transportation of water from soil into the root. We know that water is drawn by osmosis process, and the root cell will actively pump ion into cell. High concentrations of ion creates a greater osmotic pressure in plant than surrounding soil water. Water will move into cell by osmosis. There are two main factor, or there are two main characters in this point. One is water and the other one is ion. Water drawn into the cell by osmosis. However, ions, they need active transport, meaning they need energy in the form of ATP to transport them into the cell. So, because of the transportation of ions into the cell, it creates or resulted with high concentration of ion. Because of high concentrations of ion in the cell, it creates a high osmotic pressure in plant than the surrounding soil water. Therefore, it will permit and invite water to move in through osmosis easily. On top of that, the passing through the transportation of water in plant roots, they are following three different pathways. We have discussed the pathway before. They are apoplast, symplast, and vacuolar. The most important and easiest as well as the most effective way is apoplast, followed by symplast, and the last is vacuolar. Apoplast is the easiest because the root of water is passing through the cell wall only. Meanwhile, symplast, they have to go through the cell wall as well as the cytoplasm. Meanwhile, the vacuolar, they have to go through all of the plant cell structure. That's why they are becoming the most hardest and the difficult way of water passing channel. We have come to the parts where the water through xylem being pulled due to the shoot tension. This is actually to answer the first question that I posted in the initial slide, asking about whether the water is it being pushed or pulled. So now we know in plant transport system, the water is actually being pushed and pulled. Pull because of shoot tension and push because of root pressure. To fully understand the concept of pulling the xylem sap by shoot tension, the slide will explain to you. Water movement in plants not only from below via positive pressure or known as root pressure, but also by pulling from above via negative pressure or by shoot tension. Potential decreases as one moves up the sand. The word one, we are referring to the sap. Now, positive pressure from below because of root tension. Negative pressure from above is because of shoot tension. The character of pressure, whether they are positive and negative, is dependent on the load of water. If there is a load of water, they call it positive pressure. If they are unloaded or empty of water, they call it negative pressure. So, the chain of positive and negative will take charge in a circle as long as the water moves along the xylem and along the plant life cycle. The process occurs via transpiration, meaning that the transpiration will directly determine whether the needs of pulling and pushing of water to take place by root pressure as well as by chute tension. The air spaces inside spongy mesophyll are quite humid. We are now talking about the internal space of leaf where they have spongy mesophyll, as they are constantly in contact with moist cell, wall, and vascular tissue filled with xylem sap. In a normal condition, these air spaces inside spongy mesophyll in the leaf, they are quite humid because they are constantly in contact with moist cell wall and vascular tissue filled with xylem sap, because this is the final destination or the final spot from where the water travel right in the early day from the root to the stem and finally to the leaf before they escape as a water vapor into the atmosphere. On typical non-rainy days, the water potential at the atmosphere is always lower, or we call it negative, because on a typical non-rainy, there must be hot and dry. So there must be no water, less humidity. Therefore, the potential becomes negative than that of the spaces inside the mesophyll, which is always to be known with a positive potential because they will always fill with water and moist. This means the water will travel out of the stomata from this mesophyll into the area of low water potential. As one moves, we are referring the word one to the water down the plant, water potential increases. I repeat again, as the water moves down the plant, the water potential will increase because now the empty spaces will be uploaded again with the water. And we will look at the simple hypothesis of water potential in a soil and a plant system with the diagram provided in this slide. In the soil with high water potential and in the atmosphere with low water potential. In the soil, there are high water potential because there's a lot of water resources. In the atmosphere, low water potential because in a normal day, there is high temperature, non-rainy, then it becomes low humidity, therefore low water potential. In the soil with high water potential followed by in the root, it may become medium to high. To the leaf, it may become low when stomata are open. And also it depends on the transpiration rate. We will discuss about the transpiration rate determined by a few numerous factors in the next few slides. In the atmosphere, usually low, but the rate of water potential in the atmosphere, it may be changed according to the humidity level. Under the objective of xylem and transpiration, we also have to know the theory of transpiration. This theory of transpiration works closely with how the xylem conduct or transport the water. And this theory is also works closely with how the water being pushed by the root pressure and being pulled by the shoot tension. Let's look at it in detail. What is theory of transpiration? Which make the xylem works in transporting water and dissolve a minerals ion effectively. Water evaporate from cell wall of palisade and spongy mesophyll cells into substomatal cavity. We are now focusing into the leaf structure because all of these palisade, spongy mesophyll, and stomata are located within the leaf organ. Now, water evaporate from cell wall of palisade and spongy mesophyll cell into substomatal cavity. Therefore, it creates lower the water cavity and it will draw the water from neighboring mesophyll cell that has higher water potential until all the water is drawn from xylem vessel in the leaf via apoplast. It can be symplast or it can be vacuolar. So, in short, theory of transpiration is telling us about the evaporation of water from cell wall of palisade and spongy mesophyll cell into substomatal cavity before they escape into the atmosphere. And the occurrence of theory of transpiration whereby the water can evaporate from within the leaf structure out to the atmosphere are following the respective factor, which is lower the water cavity and the drawing of water from neighboring mesophyll cell that has high water potential, and this water are being drawn from xylem vessel in the leaf, where originally all of this water are from the stem, are from the roots, are from the soil. Now, in theory of transpiration, there are two factors which support this theory of where the water can actually move from one space to another until they escape to the atmosphere. The factors are cohesion and adhesion. What the cohesions do in theory of transpiration? The water molecules will form continuous water column in the xylem vessel by hydrogen bond that form between the water molecule. Meanwhile, the adhesion is about the attractive forces between water molecule and the hydrophilic xylem wall, which will prevent the water column from moving downward. This is because in xylem, there should be only the moving of one direction upward of the plant. Therefore, here they come, the purpose and the function of adhesion to prevent the water column from moving down. Now, we know that by having those character, by having those principles, theory of transpiration is fully supported by cohesion and adhesion force within the water and the hydrophilic xylem walls. Let us recap how the cohesion and adhesion support the theory of transpiration. In cohesion, the polar water molecule that will tend to stick together with their existing hydrogen bond. Meanwhile, in adhesion, the water molecule will stick to the hydrophilic xylem wall surfaces. And this cohesion and adhesion actually lead to what they call a capillarity reaction of water in xylem, where due to adhesion between water molecule and the hydrophilic surfaces of the wall of the narrow elongated xylem vessel and the cohesiveness forces which hold the water molecule to each other in the water column, that makes the capillarity reaction of water movement along the xylem vessel. Now, more water loss, more water is continuously pulled into mesophil out to the atmosphere, and these forces are called the transpiration pull, which pull the water more to come into the mesophil or the internal air spaces in the leaf before they leave the leaf via the stomata. And we know now the cohesion, adhesion, forming capillarity reaction forces that help the water to move along the xylem vessel. From the previous slide, I have shared with you a theory of transpiration. We have known how the cohesion adhesion tension which form the capillarity reaction which moving the water through the xylem vessel. And this theory of transpiration, they actually creates a water pressure gradient. The transpiration itself creates a water pressure gradient. When we look at this slide with the diagrammatic features showing, we know that root pressure with a positive hydrostatic pressure will push the water up the stem. This positive pressure in the root is because of the load of the water in mass. The load of the water in the root is full. That's why they become a positive pressure. When it goes to the next level, water will move from root cell, drawn into xylem, and it will produce the root pressure. Because of the root pressure, the water can actually crawl and move from the root cell into xylem. And as water moves from one section, from one segment to another, the water potential decreases. This is following whether the space, the sections, or the area is loaded or empty. If they are loaded, they are positive. If they are empty, they are negative. Until they reach to the endoderm cell and subsequently or consequently will actively secrete those water or even the dissolved minerals ion into the xylem until they pass through to the other parts of the plant which require the water to be transported to. And when we look at the diagram provided in this slide, we can see the value of water pressure at soil, root, trunk or stem, leaf, as well as the atmosphere or the outside air. The pressure or the gradient is actually increasing. I mean, it decreases as the water moves from the soil to the root, next to the stem, next to the leaf, and finally to the outside environment. We know that there are low potential at the top. It is because the tension that pull water up from the bottom. When the lower potential at the top, this is becoming the big reason the water to being pulled from the bottom. And now let's look at the transpiration process which create the potential gradient throughout the plant. Let us begin with the top part of the plant, which is the leaf. We know that the leaf can also create a specific pool, which they call transpirational pool, which will draw the water from the neighboring cell of the stoma to to the stoma from the mesophyll cell. These mesophyll cells are originally from the palisade, and these palisades are originally from the xylem, and these xylem are originally from the stem, from the roots, and from the soil. And now, water which evaporates through stomata of leaf, and evaporation will take place at the surface of the leaf, which will keep the water column moving. And this is the strongest force which involved in transpiration to occur. Okay, we know that when water evaporates through stomata of the leaf, especially on the aerial parts of the leaf, then this evaporation will keep the water column moving, and it becomes the strongest force in the transpiration. When the water moves out to atmosphere through stomata, therefore it becomes the strongest force that pulls the water from the neighboring cell. Now it comes where the cohesion of water molecule to one another and adhesion of the water to the hydrophilic xylem wall by the existing hydrogen bond will create a water chain. So now we know the cohesion and adhesion which form the capillarity reaction, will cause the water to crawl up into the narrow tubes of the xylem. And the narrower the tube, the higher the same mass of water can climb up the xylem vessel. And on top, cohesion is the effect of water molecule which stick to one another, creates a water chain effect. And as the molecule remove from the column by evaporation in the aerial surface of the leaf, more water will drawn up through this xylem vessel. Next is the process where the water are being absorbing from the soil environment into the root cell. This is because originally the water escaped the plant through the leaf, through the stomata, outside to the atmosphere, and it creates the strongest force which will draw the water from the water column of xylem in the stem. And when the water leaves the water column, move to the leaf, therefore the empty spaces in the water column need to be replaced. And the replacement will be conducted by the water movement through the soil into the roots, and the subsequent effect and the subsequent change will take place. We know that the pressure differences created by transpiration in the early of the process will draw water out of the roots and go up the stem. And this creates a lower water pressure in the root, which will draw in more water from the soil environment. Therefore, the effect of pressure differences, the effect of concentration gradient, they are all helping one to another to support the transpirational pool, to support the capillarity reaction, to support the drawing of water, as well as the expelling of water in and out of the plant. These diagrammatic features is showing you the exact general structure of the internal leaf. The cross section of this is showing the location of mesophyll, the location of air spaces, the location of stoma, where is the cuticle, where is the epidermis, as well as the xylem. We know that leaf structure and also other parts of the plant are being covered by a waxy protective layer called cuticle in order to help them to prevent from drying out, because we know plants are exposed to high temperature, exposed to the sunlight. Therefore, they need a special character to prevent themselves from drying out. Therefore, it comes the cuticle to take this role. And on top of the leaf, we have upper epidermis, and below we have lower epidermis. And the exact location of stoma is under the lower epidermis. In between the upper and lower epidermis, there are mesophyll, including the palisade. Palisade is close to the upper epidermis, and mesophyll is close to lower epidermis. And within the mesophyll, there are a huge spaces of air. These huge spaces of air is actually function as a preparing to receive more water to come in before they escape to the atmosphere through stomata. And in this slide, or in these diagrammatic features, we can also see location of xylem. Actually, in the whole plant, xylem, they will end at the leaf and they will begin at the root hair. So now you know xylem is not only in the stem of the plant, but they are extensively long, narrow and hollow structure right from the roots until the leaf. That's why the transportation of water will take place efficiently throughout the plant system because they can be transported along the way from the root to the leaf. In this slide, we will discuss about the water molecule when they reach in the mesophyll cell in the leaf. Now, we know that water will follow the potential gradient from soil to atmosphere, and they are being pulled together by the cohesive and adhesive tension of the molecule itself. The negative pressure in the xylem tube will generate it this way, which means producing shoot tension. As I've mentioned to you before, shoot tension has been created with negative pressure because the situation of where the water is empty. Therefore, the empty means negative pressure. The positive means with full of water. And water will diffuse from xylem into the spaces inside the spongy mesophyll, next to the stomata, and finally to the atmosphere. And the water which crawl into the xylem vessel and finally into the leaf, they are actually will coat or coating the surface of the mesophyll spaces and form crescent-shaped menisci as it enters the air spaces. As water molecule pull from between the cell, the menisci become more curved and water tension increase due to the cohesiveness of the water molecule. Because we know water molecule that tend not only to stick with the other water molecule, but they also tend to stick with the other hydrophilic wall in the plant, including this mesophyll cell. The meniscus tension is inversely proportional to the radius of its curved surface. As the meniscus becomes more concave, the radius will decrease. However, the tension will increase. As what we can see from the diagram in this slide, the meniscus of water formed by pulling off the water molecule into the air spaces, and it will create the concave shape together with the changing in the meniscus formation. And when the meniscus become more concave, the radius of this meniscus will decrease, and at the same time the tension will increase, because at the same time these water molecules are also attracted to the hydrophilic wall of the cell. But in overall, we know water, they are following the potential gradient from soil to atmosphere, and these potential gradients are helped to crawl of the water by pulling them together into the formation of cohesive adhesive tension of the molecule themselves. And the other factor which create the transportation of water in the theory of transpiration of what they call shoot tension. So, in conclusion of transpiration, it is a process where loss of water, as in the form of water vapor from plant to the atmosphere, up to 99% of all water amount from which the plant absorbed in the early days. And more water is lost through stomata. There are some through cuticle layer, and there are also some through lenticule, especially in higher level plant, which known as woody plant or woody stem. Only 1% of the absorbed water are being used for photosynthesis or other metabolic processes, and the rest, out of this 1%, they are also used it to maintain turgidity and freshness of the plant cell. And rate which are regulated and control the transpiration rate are done by gut cell which surrounding the stomata. We will discuss and we will see how the gut cell control the opening and closing of the stomata, and finally determine the rate of transpiration. And gut cell, they will open when water moves into cell by osmosis, then create turgid. The turgid results from active uptaking of potassium ion stimulated by light and the subsequent influx of water by osmosis. This event normally takes place during the daytime. In this slide, we can see the microscopic features of stomata, where the stomata is composed with two gut cells on the left and right sides, becoming the opening and closing channel to allow or to prevent water to escape from the cell out to the atmosphere. And we know that this stomata, which made up of two gut cells, they can be open and closed. Normally, it depends on the light received and also the rate of water movement. In order to maintain the capability and the capacity of the stomata in order to perform the opening and closing to release or to prevent water leakage, they are a special structure called cellulose microfibril, which can prevent cells getting wider when they swell. This is especially when the water influx and it creates the cell turgor. Therefore, this cellulose microfibril will become a prevent measure to retain the optimum and correct size of cell turgor. It will not go beyond the excessive size, or else it will damage the cell. And here we can see factors of moving in potassium ion as well as the water molecule will affect the formation and also the shape of the gut cell. Normally, the water will enter into the gut cell by osmosis following water potential gradient from high to low. And gut cell will also actively pump the potassium ion in order to make the stomata to open during the daytime. At night time, this potassium ion will actively pump out so that it will become flaccid and closed. This will be another recap for us to see how the formation of opening and closing of stomata controlled or regulated by two gut cells. On the left side, we can see the opening of stomata, and on the right side, we can see the closing of the stomata. Both are controlled by two gut cells. And we know when waters come in, it will make the gut cell swollen, turgor, and it will open up the space for water to escape. And when the water influx out, it will make the two gut cells flaccid and return to the normal size. Therefore, the stomata will close and prevent the water from flowing out as in the form of water vapor. In open stomata, solutes potential is normally high and water moves into gut cell. Meanwhile, in closed stomata, solute potential is low. Therefore, the water will move out of the gut cell. Here we come to factors which affect a rate of transpiration. By looking at the factors that I've listed here, we could say that they are all considered as abiotic factors because they are non-living factors which affect the rate of transpiration. We know that in order for the plant to conduct or to perform transpiration, there are many other elements which contributes into the process. The process are, or the elements are, namely the shoot tension, the root pressure, the transpirational pool, the theory of transpiration, cohesion, adhesion, tension, capillarity movement of water, concentration gradient, membrane gradient, and many more. But apart from that composed of element, they are also factors which affect the rates of transpiration. And they are temperature, light intensity, humidity, as well as the wind speed. Let's look at it one by one. For temperature, as the temperature increases, the rate of transpiration will also increase. For the light, as the light intensity increases, the rate of transpiration will also increase. For wind speed, as the wind increases to blow, the rate of transpiration will increase as well. However, for humidity, as the humidity increases, but the transpiration rate will decrease. When we look at to all of these four factors, the three of them are directly proportionate. Meanwhile, the other one is indirectly proportionate. We can say that for temperature, light intensity, as well as wind speed, they are positively correlated with rate of transpiration. Meanwhile, for humidity, it is a negative correlation with rate of transpiration. What is so important about having transpiration in plant? The purpose of having transpiration in plant is to maintain water potential gradient that moves the water and any dissolved minerals from root to any aerial parts of the plant before they escape as in the form of water vapor to the atmosphere. And this evaporation of water from leaf, it will also absorb the latent heat of vaporization and therefore cooling down the leaf during hot and dry weather. These are all the contributions mechanism to maintain in order for the plant to endure with the open air spaces, to endure with exposure to the sunlight every day. Okay, now, before we end this section, let us recap the factor which determine the rate of transpiration in further detail. Factors which can determine the rate of transpiration can actually be divided into two major factors, which are the external factor as well as the internal factor. Factors of external, we have temperature, light, humidity, air movement, as well as water supply. Meanwhile, in internal factor, we do have leaf surface, locations of stomata, and finally the density of stomata pore. Now, in temperature, increased kinetic energy will increase movement of water molecule. Therefore, it will diffuse through stomata faster, meaning that as the kinetic energy increase, the movement of water molecule will increase, then they will move faster through the stomata. Under the high temperature, low humidity, it will increase the rate of diffusion in the form of water vapor from leaf to the atmosphere. What about light? During the daytime, with high light intensity, it will stimulate the opening of stomata. Therefore, the transpiration rate will increase. At night time, during low light intensity, it will stimulate the stomata to close and therefore the transpiration rate will decrease or reduce. In humidity, low humidity translated into high vapor pressure. Therefore, with increased gradient of water vapor saturation between substomata cavity and atmosphere, it will increase transpiration. In short, low humidity increased transpiration. In air movement, normally the air movement will carry away water vapor from outside stomata. Thus, it will create steep concentration gradient and the water vapor from stomata cavity will diffuse out rapidly. In high, dry, windy condition, definitely the transpiration rate will increase. Under the low air movement, therefore, the water vapor will accumulate around stomata and it will reduce the concentration gradient. Thus, it will reduce the water loss out of the leaf to the atmosphere. And the final factors, under the external factor, we do have water supply. Now, rate of transpiration is higher than the rate of water absorption from soil. Therefore, the stomata to close and it will reduce water loss by transpiration. In this factor, we have to understand that as the water supply into the plant is low, therefore the plant has to do a mitigation or the action to reduce the transpiration rate by closing the stomata to reduce the water loss of transport. In internal factors that determine the rate of transpiration, leaf surface, bigger leaf surface, higher the transpiration rate. Less stomata, thin needle shape and roll leaf will reduce the surface area exposed to the air, therefore result with reduced evaporation of water from leaf. Location of stomata. Normally we found lots of stomata under the leaf area. They are not on the upper part of the leaf. The creation of stomata under the leaf area is to reduce the loss of water naturally. And finally, the density of stomata pore. The bigger the density, the more the water loss. And before we move on to the final section of this objective, I would like to recap once again about xylem and transpiration, how the process occurs. We know that the movement of water is up the xylem vessel is in the form of mass flow, and the water column is always under tension and is pulled from root to the leaf. And this process will reduce the hydrostatic pressure. Why? Because water is moving from one segment or one location from one to another. And finally, the water from top of the xylem vessel will reduce because they are now transported into the leaf before the final destination to the atmosphere in the form of water vapor. Talking a lot about functions, role, and how the xylem conduct the water throughout the vascular stream. But what makes the xylem is so special, relevant, significant, efficiently and optimally conducting the water transport in plant? Thus, what are the characteristics of xylem vessel in facilitating water transport in plant? We will scrutinize. There must be a special features of xylem in conducting the water effectively. This is the characteristic of xylem vessel that are we talking about. There are 6 in total special features of xylem vessel. We will start with they are hollow and tubular cells. Number 2, they are composed of 5 cells type, including trachea, vessels, parenchyma, sclerids, and fiber. Each of these 5 cell type will definitely doing their part in supporting the xylem vessel to withstand. The trachea, for example, they are small in diameter, and vessel, They are large in diameter, form the bulk of the tissue. So the difference between trachea and vessel is just the diameter of this structure. They are heavily strengthened and are the conducting cells of the xylem. So now we know the one that strengthening the xylem is played by the trachea and also the vessel. What about parenchyma, fibers and sclerae? Parenchyma, they will act as a storage space. They will act as a place to store the food, to supply, in biologically, they will act as a store to keep the food as for a nutritionless cell. Fibers and sclerae, they provide the support. To be specific, the mechanical support for the xylem. The xylem with death at maturity. With no protoplasm, however, they will form a hard skeleton that will serve only to support the plant, meaning that even though at maturity, at mature state, the xylem will death, however, the death will not regenerate and lost just like that. They will becoming a hard skeleton that serve to support the plant. The xylem vessel also lignified on their cell wall. This is in order to give strength to withstand the tension and to prevent them from collapsing and makes the xylem waterproof. Vessel does not collapse under tension and water does not seep out. That is the purpose of lignification of the xylem vessel cell wall. We can see by lignifying the cell wall of xylem, this lignification will actually to give strength by withstanding the tension and preventing the vessel from collapse. Because we know xylem, they have to conduct water transport throughout the plant all over the time. Other than that, lignification also will ensure the waterproof as well as the vessel will not collapse under tension, and by waterproofing, the water will not seep out, so it will move in one direction without any difficulty and less efficient. They are also long continuous tube. They need to be long and continuous because the plant structure can be long or very long. They must be continuous tube because in order to ensure the efficient water transport without any barrier. And the last character, they are narrow and capillarity. This is to increase the adhesion between water molecule and cohesion between water molecule and hydrophilic cell wall of the xylem. So here we can conclude that 6 characteristics which help the xylem to become effective water transporter, including hollow tubular cell composed of 5 cell types including tracheae, vessel, parenchyma, sclerae, and fiber. The death at maturity, but the death will become a hard skeleton that serves to support the plant mechanically. They are also lignified cell wall to give strength to provide the waterproof. They are also long and continuous because to be significant in transporting water as well as to speed up the movement without any barrier. They are also narrow and capillarity in order to push the water up and to pull the water up. In this slide, I am sharing with you the microscopic structure as well as the diagrammatic structures of the xylem once again. We can see where is the vessel, where is the trachea, where is the vessel member. We know that vessel member are supported by trachea in order to ensure the xylem vessel to withstand storm to conduct water transport. And within the vessel as well as the trachea, we can see there are another additional features of them, where we can see on the cell wall of the vessel they are pitted in order to allow the transportation of water from one vessel to another vessel without any barrier. And within the vessel itself, we can see there's the pore. The pore is to avoid any barrier of water transport from one vessel to another. This will be the final slide under objective number 1. The further classification of xylem into protoxylem and metaxylem. But you do not need to be worried about this slide. This will be additional information that I wanted to share with you about the classification of xylem. Now, they are protoxylem, they are metaxylem in the form of differences of character and shape. Protoxylem, they are annular and spiral thickening which allow the stretching of walls and further elongate the stem is possible. Meanwhile, metaxylem, they are clariform, reticular and pitted which thickening provide the additional mechanical strength to all the stem. But both are functions in the same way to transport water. The difference is just the way they are formed. One with annular and spiral thickening, another one with scalariform, reticular and pitted thickening. But both function similarly in providing a good transportation system of water. In terms of size, metaxylem vessel, they have larger lumen than the protoxylem. Therefore, they will transport more water up the plant. So, if the trick question asking you which of the xylem is better option to transport water, there must be the answer of metaxylem, because metaxylem, they have vessel with larger lumen, bigger than the protoxylem, but both function to transport water. It's just that metaxylem vessel can transport more water up the plant compared to the protoxylem. And within this slide, you can also see the location of protoxylem as well as the metaxylem in the microscopic features above below, where you can see in the red stain of the microscopic features showing the protoxylem and also the metaxylem. They are distinguished by the size of the lumen or the size of the vessel.